Hello, my name is David Murray. I'm the Associate Director for Prevention and the Director of the Office of Disease Prevention at the National Institutes of Health. I'm pleased to welcome you to the last of three 2026 Early Stage Investigator Lectures. We created this lecture to highlight early career prevention scientists who have not yet served as a principal investigator for a substantial NIH-supported research project, but who have made outstanding research contributions to their respective fields and are poised to become future leaders in prevention research. Before we begin, I have some housekeeping items. You can submit questions during the webinar using the Q&A pod. You'll need to open the pod by hovering your cursor at the top of the Teams window and then clicking on the Q&A icon. We will open the floor to questions at the end of the lecture. The video recording of the lecture will be posted on our website in a few weeks. We're excited to present our final 2026 awardee, Dr. Meichen Wang. Dr. Wang is an assistant professor in the Department of Environmental Health Sciences at the University of Massachusetts Amherst. She earned her master's and PhD degrees in toxicology from Texas A&M University and completed postdoctoral training on environmental remediation funded by the National Institutes of Environmental Health Sciences, Superfund, and K99 programs. Dr. Wang's research program focuses on developing edible clay-based sorbents that mitigate dietary exposure to per and polyfluoroalkyl substances, also known as PFAS, and prevent their toxicities during critical windows, such as pregnancy, lactation, and early development. Her work has resulted in 50 peer-reviewed publications, a patent for clay technologies, and a license for technology transfer. Her work aims to bridge fundamental toxicology and practical interventions to establish clay sorbents as a primary dietary barrier that reduces internal chemical burden and prevents chronic disease. In today's lecture, Dr. Wang will discuss how dietary intake represents a major route of human exposure to per and polyfluoroalkyl substances, and how effective interventions to mitigate this exposure are limited, especially during vulnerable periods. Her interdisciplinary team employs a combination of absorption analyses, in vitro cell models, bioassays, and rodent models to validate the efficacy and safety of the sorbent strategy to protect human health from these persistent contaminants. Please join me this morning in welcoming Dr. Wang.
Thank you, Dr. Murphy, for the kind introduction, and thank you all. Thank you all for attending my talk today. I am greatly honored to be here and share my research and its implication for disease prevention. But let me start first with my research journey. Before joining UMass as a faculty, I did my PhD and postdoc in Dr. Phillips' lab at Texas A&M University, and we were funded by the NIEHS Superfund program, and we were working on the remediation engineering project. So we developed intervention strategies using sorbents like carbon and clays. They act like a sponge that can grab and immobilize chemicals. And I will briefly go through 4 primary projects as examples to show you how we remediated chemical contaminants in water, soil, and air. So a key focus of my work has been on the persistent organic pollutants, which could accumulate in soil. And people can get exposed to soil through thermal contact, hand-to-mouth behavior for children, and crops and plants grow on contaminated soil. So to address this problem, we developed sorbents as soil supplements for community garden and lawns. And those sorbents, when they're included in the soil, they can sequester and immobilize chemicals particularly pesticides and PFAS in the soil, and thus reduce their leaching to groundwater and uptake by agricultural crops. These lipophilic chemicals also tend to be found in the sediments and accumulate in fish and shellfish, and oyster is one of the most prominent form of marine aquaculture in the US. And they are vulnerable to water and sediment pollutants because they are filter feeders at the bottom. So this project showed that when we added sorbents to the water and in the feed of the oysters, they bind PCBs and thus reduce PCB residues in the edible parts of the oysters and improve the food safety. So this sorbent material was patented partially because of this study and this application. Another direction we have taken is to develop a sunscreen with sorbents in it as a physical barrier to block the penetration of chemicals to the skin. So this is a critical need because you see first responders, they walk in flood water or in heavy rain, and they can get thermal exposure to chemicals and bacterial infection in the water. So we wanted to use this heavy and thick cream with sorbents in it as a physical barrier together with PPE to block thermal contact. Lastly, we have incorporated sorbents in air filter inserts in face masks and air conditioning to reduce inhalation exposure to volatile organic compounds. The results showed that the sorbent-loaded filters, they could trap benzene in the air and thus reduce benzene indoor concentrations. So I know I run through these projects fairly quickly because I just want to use them as examples to show you my background and the versatile use of sorbents in environmental remediation and reflect my commitment to solve real-world problems through material innovation. And you can see that I have done research in different directions in environmental remediation, which guided me to switch my current research focus to protect human dietary exposure to environmental chemicals from food and drinks. So why do I zoom in onto the dietary exposure? Because for average American adults, the daily food intake is almost 6 pounds, and it is recommended to drink half a gallon of water per day. So food and water safety are urgent priorities. And the hot zone in recent decades has been doubled, where the risks of contaminants are higher. So now affecting 4.5 billion people and half of North America in this hot zone. So what can we do to protect ourselves? Have you ever thought about eating clay could be a solution? So the goal of my research is to solve dietary exposure using edible clays as toxin binders. When these clays are included in the contaminated food and water, they can immediately grab and sequester chemicals in the food and water and continue desorption when we ingest them in the GI tract. And because of those are clay materials, our body won't uptake them. So they just carry the contaminants and pass through the body directly for direct excretion, preventing chemical uptake in the body and also prevent the downstream toxicity. And our target is to protect populations, especially during susceptible windows of exposure, such as pregnancy, lactation, and the developmental stage of newborns. So why do we want to solve the problem using clays? It's because human ingestion of clays or geophagy has been observed since the earliest record all over the world as ancient medicine to treat diarrhea. It's also a culture and culinary habit to eat clays, even today, especially in Africa, Middle East, and South America. So our hypothesis is to solve the problem by developing edible clays as toxin binders to reduce environmental chemicals, pollutants in the food and water. And the base clay, the parent clay material, the unamended clay material we use is called montmorillonite. It is generally recognized as safe material, and animals fed with this parent clay has not only show minimal impact on the gut microbiome, but also to help with their digestion and also stabilize their intestinal conditions. And based on Phillips' 6 human clinical trials in Africa and Texas, this parent clay has been shown to be safe for human consumption without interference with nutrients. And not only is it safe, this parent montmorillonite clay was also shown to be able to reduce foodborne aflatoxin biomarkers in the humans. And because of the studies, this parent clay has been included in about 20% of the animal feed in Europe. But this parent clay is only selective for certain hydrophilic chemicals like aflatoxin. But because dietary or oral exposure is a major exposure route for many environmental chemicals, because the environment is a solvent for food, resulting in complex mixtures of natural and synthetic pollutants from sediment, water, and air. They could all end up in our diet. So we are what we eat. So there is a critical need to mitigate this dietary exposure to environmental chemicals. And one chemical with major dietary exposure and health concern is PFAS, also known as the forever chemical. They have emerged as contaminants of global concern because of their potential to accumulate in the human body and the food chain. Actually, PFAS has been detected in most of the US populations tested, and the PFAS contaminated food and drinking water accounts for more than 90% of human exposure. PFAS also goes through this process called enterohepatic recirculation, meaning that the ingested PFAS, after they are absorbed by the liver and by the bioassay, they could be kicked back to the gut. So this recycling process could prolong PFAS gut residence and cause repeated systemic exposure, cumulative stress, and increase their risks of chronic disease. However, there is currently no clinically approved orally deliverable interventions that block its gastrointestinal uptake or to mitigate the systemic accumulation. So my study aims to address this critical gap by developing a gut-targeted strategy that intercepts PFAS at the point of ingestion. So my current research is to, on top of this parent-based montmorillonite clay, I added about 30 different kinds of materials on it, such as nutrients, probiotics, prebiotics, to develop new edible sorbents that could target dietary PFAS. And to characterize how well does this newly developed clays can react and remove PFAS, I used a combination of methods, including in vitro adsorption studies in the simulated gastrointestinal tract model to look at a series of interaction parameters, such as how much PFAS can be bound, how long are they remain binding, and how fast is the binding, etc. And these parameters are validated by different cell lines for the reduction of cytotoxicity after clay treatment, and also to validate PFAS toxicity pathways after PFAS is removed by the clays. Also, chemicals in the clays, they are exposed to living organisms that are sensitive to the environment and thus multiple endpoints such as their growth or reproduction, they're used to indicate toxicity of PFAS and also detoxification after clay treatment. Lastly, we use in vivo rodent models to validate the safety and efficacy of eating sorbents in reducing toxicity. So in today's presentation, the results will mostly focus on two different edible clays, which are montmorillonite clays amended with carnitine, which is a amino acid derivative, and also montmorillonite clay amended with choline, which is a vitamin-like compound. So we first conducted adsorption exotherms and thermodynamics in the stomach model to characterize what's the surface chemistry of clays to interact with PFAS. And you can see in the figure, the results show fit this Lamure model showing a high binding capacity. So that's the maximum binding available, showing where it saturates in the plateau, showing that there are sufficient binding sites available on the clay. The figure also showed high binding affinity, this angle of slope, showing that the clay is sensitive to grab PFAS and the reaction is fast. I also collapsed the clay so that the clay layers, they stack on each other without the empty space, like a deck of cards. And you can see the less binding onto the collapsed clay, suggesting that this empty space, the empty space between the two clay layers, that's the major binding site for PFAS. Also in the thermodynamic studies, we observed the high free energy absorption and enthalpy values that indicate tight binding of PFAS. That once they are bound, they are not easily dissociated, which also you can see, which explained this low desorption rate, less than 12% in the intestine model. So to validate these experimental results, our collaborator, Dr. Tamamis, used in silico computational modeling. So in the simulation, we saw the enhanced binding of PFAS on the carnitine and choline, those amended clays compared to the parent montmorillonite clays. And also the major binding mechanism that accounts for 60 to 90% of the PFAS binding was through electrostatics and the hydrophobic interaction. So those are chemoabsorption, explaining the tight chemoabsorption that we just observed on the last slide. So after PFAS is ingested in the gut, they could reach to their target organs and is believed to associated with all kinds of disease. And one area I'm particularly interested in is its role in metabolic disorder, because epidemiological studies have associated PFAS exposure with increased risk of obesity and BMI, suggesting these chemicals may disrupt lipid homeostasis. And to test this hypothesis, and also to explore whether clays could mitigate this toxicity, I used a pre-adipocyte cell model called 3T3L1. So this cell model could be differentiated into mature adipocytes, and that's why it's widely used to test metabolic disease. And the results show that the PFAS exposure during the 10-day differentiation process showed dose-dependent toxicity, and also the toxicity is time-dependent and also additive, that the PFAS mixture containing 4 individual PFAS, including PFOA, PFOS, GNX, and PFBS, the mixture is more toxic than individual PFAS. But the good news is that after reacting with clays at 10 to the minus 5 to 10 to the minus 4 doses in the cell media, those clay treatments dose-dependently improved the cell availability in the MTT and LDH assays during this differentiation stage. So we further exposed the cells with PFAS at 0.01 micromole for 10 days during the differentiation stage. And we see that at PFAS at this relatively low dose increased the production of ROS and nitric oxide, which, you know, the excessive amount of those chemicals could induce oxidative stress and inflammation. PFAS at this level, 0.01 micromole, also disrupted mitochondrial membrane potential as suggested by the GC1 dye, showing they could interfere with mitochondria and potentially affect ATP production. But importantly, the clay treatment, again, at 10 to the minus 4 to 10 to the minus 5 levels in the media, they reduced this cytotoxicity, correlating with reduced PFAS uptake within the cells. So PFAS after 10-day exposure, we see that about 7% to 36% of PFAS were detected in the cells after 10-day exposure. And after clay treatment, whether it's the parent amended clays or amended with carnitine, with choline, or chlorophyll, they could all reduce the PFAS exposure levels to different extent, explaining the reduced cytotoxicity. Also, for differentiated adipocytes, so the fat cells, they store energy primarily in the lipid droplets, which are stained in red. But you can see that the PFAS exposure during the differentiation, they increased the lipid droplets by 2 to 4-fold, possibly by increasing the glucose uptake within the cells by up to 4-fold. So these findings imply that even this relatively low dose PFAS level could interfere with adipocyte function by increasing glucose uptake and fatty acid synthesis, and driving like metabolic disorder linking obesity. And by looking at the expression of genes relevant to inflammation and metabolism and fatty acid, we found that certain gene expressions are highly correlated with PFAS cytotoxicity, including TNF-alpha and IL-6. Those are pre-inflammatory cytokines that are correlative with the cell viability toxicity. We have also seen the gene expression in INOS/ARG1. They are highly correlative to the nitric oxide production, as well as the PPAR-alpha expression highly correlates with the lipid droplets amount. So by integrating the mechanistic toxicity with mitigation intervention, this work firstly validated that the PFAS adipogenetic toxicity is through oxidative stress, metabolic disorder, and inflammatory pathways, and also suggests that this nutrient-amended clays could be used as sorbents to reduce PFAS bioavailability and thus prevent their toxicity pathways during the adipocyte development. Furthermore, to test the safety and detoxification efficacy of these clays, a battery of bioassays were used in my lab, including Hydrovulgaris, which is a cnidarian just like jellyfish. It is the highest organism capable of self-regeneration, and it is asexual proliferation, meaning they are genetically identical. Lamina minor, also known as duckweed. It's a floating plant and it's recommended by EPA for pesticides regulation. And also C. elegans is a soil nematode, and it has emerged as a powerful model for studying obesity and metabolic dysfunction. Because of its highly conserved lipid metabolism pathway that they share about 65% of human disease-related genes. They also have transparent body for direct visualization of lipid accumulation and short life cycle. So these models are sensitive to environmental chemicals, and thus they're used as toxicity indicators. So some of these results are produced by my student Danish, and our results in the hydro assay showed dose-dependent toxicity that PFAS at high dose reduced the PFAS morphology from score 10 at the beginning of the study, where hydra is this healthy and extended morphology, reduced it to score 0, which means completely dead and disintegrated hydra within 7-day exposure. PFAS has also shown to inhibit hydra's ability to regenerate itself. That you can see compared to the vehicle control. If we cut off the head and the bottom of the hydra, they were able to regenerate itself to the full body within 7 days. But PFAS exposure at levels higher than 50 micromole, they completely inhibited this regeneration ability. PFAS at low dose also increased the ROS production, but because at high dose, they killed the hydra, so the ROS production dropped to 0. And the PFAS, even at low doses, even if they didn't kill the hydra completely, they were shown to interfere with their ability to prey and consume feed, suggesting that the hydra could be sensitive to PFAS toxicity. Similar results are shown in C. elegans, that PFAS levels, they dose-dependently decreased the C. elegans, reduced their survival rate, reduced their growth, as shown by reduced body length. PFAS has also shown some neurotoxicity in the C. elegans, as shown by the reduced head thresh. That's how they move around, reduce their pumping rate. That's how they consume feed, and also reduce their locomotion. That's how they respond to environmental stimulus. And lastly, PFAS also reduced their reproduction by laying less eggs. And lastly, PFAS also increased the ROS production, just like what we saw in the Hydra vulgaris model, as well as in the cell model. But the good news is that with the inclusion of clays at 0.01% and the 0.1%. Those clays in the hydromedia showed 25 to 80% of protection in hydromorphology and regeneration against this PFAS mixture toxicity. And also in the C. elegans model, the inclusion of clays at 0.5% and 0.1% in the culture media, they improved the C. elegans neural behavior, like in the locomotion, pumping rate, and head thrush, as well as restored their body length reproduction and ROS production. And those reduced the toxicity in the clay treatment groups also correlated with the reduction in the PFAS exposure, PFAS levels in the C. elegans, that we see different clay treatments, they almost completely wiped out long chain PFOA and PFOS in the C. elegans, where moderately reduced the short chain JNAX and PFBS, explaining the reduced toxicity. So right now, this is an ongoing study because we're looking at more metabolism-related endpoints in C. elegans, including triglyceride and the glucose uptake. So although PFAS is widespread and most people are exposed to PFAS, major health risks are primarily associated with prolonged or high level of exposure, such as communities near industry sites or firefighting training sites. So I'm not saying we should be panic or anything, but people could become more susceptible to PFAS toxicity during critical windows, such as pregnancy and lactation and development of newborns. And despite the risks, studies on effective strategies to reduce internalized PFAS exposure and during the critical windows is limited. So to address this gap, we exposed male and pregnant female Sprague dolly rats with both a PFAS mixture as well as clays at 2% in the oral gavage for 7 weeks during the pregnancy and lactation. And what we found is that PFAS exposure, they reduced female body weight gain during the pregnancy. They also increased the liver weight and increased the liver size. And that may be related to higher hepatic lipid accumulation, like what we saw in the cell model. PFAS has also, decreased the serum cholesterol, decreased total protein, globulin, phosphorus, and glucose, indicating PFAS toxicity in the liver and the metabolism. We have detected PFAS in the liver and the serum of both female and the male. Specifically, we have seen accumulation of PFOS that is 3 to 5 times higher than the PFOA concentrations and higher than the short chains, especially in the liver, higher than in the serum, because liver is the major detoxification and metabolism organ. We have also seen higher PFAS levels in the female liver than the males, maybe because the female, the absorption rate in female is shorter than the males. So for example, the females take 1 hour to uptake PFOA versus 10 hours in the males. But importantly, clays, as we can see in this different colored boxes, different clays, they all reduce the PFAS concentrations in the liver and serum by up to 74%, especially, the green and orange bars, those are Carnitine and choline clays, really significantly reduce the PFAS concentrations in the liver and serum. So following uptake in the blood and liver, PFAS showed hepatotoxicity by causing histopathological changes to the liver with cell apoptosis and inflammation. PFAS has also affected the antioxidant and inflammatory markers, as you can see in this paragraph. I'm not going to read it, but those changes collectively indicate PFAS-induced systematic inflammation and liver damage. So those results align with the literature review that I've seen a lot of papers showing PFAS hepatotoxicity. But what I think is a little less reported is that PFAS has also affected vitamin levels in the serum and liver, possibly by inhibiting the vitamin synthesis and accelerating their degradation. But importantly, clay treatment in the oral gavage reduced the hepatotoxicity in the parameters shown in those paragraphs, and we didn't see any interference with vitamins, but even those clays, they restored the reduced vitamin levels caused by PFAS. So for now, we only detected two vitamin levels in the rodent studies, and we have identified third party and other collaborators who could do a whole panel of nutrients that we can analyze exactly if clay interfere at all with any nutrients. And if there is any significant nutrient loss, we could calculate the amount and provide a separate dietary supplement to compensate if there is significant loss. But other than hepatotoxicity, we have also looked at PFAS toxicity in reproductive organ in the rats because, PFAS is a known endocrine disruptive chemical. Specifically, we have detected PFAS accumulation, especially PFOS, at a much higher concentration in the ovaries and testes. But the good, and also PFAS showed toxicity by altering the histopathological structure of ovaries. But importantly, clay treatments reduced PFAS bioavailability in the ovaries and testes by up to 50%, and thus reduced their toxicity. These results show that clays, orally delivered clays could reduce prenatal and postnatal exposure to PFAS in adult rodents. But the story doesn't end here. PFAS not only affected the adults who were directly gavaged with PFAS, we have also detected PFAS in the breast milk, and especially PFOS, that is time-dependent accumulation. And as a result, we actually detected about 1% of PFAS in the milk were transferred to the serum of the offspring, and about 3 to 6% of the PFAS detected in the milk was transferred into the offspring livers. And that, offspring has been indirectly exposed to PFAS through breastfeeding. And we have observed that offspring in the PFAS exposure group, they showed reduced later birth weight and reduced growth by 21% compared to the vehicle control by the end of their breastfeeding period. And the inclusion of clays showed some tendency to reverse this toxicity, although the detoxification is not as significant as in the adult because the offspring, they were not treated with clay or PFAS, but we did see the tendency in the offspring as well. So collectively, we're really excited to see that our results from in vitro study in the simulated GI model showed high binding and tight binding of PFAS onto the clay surfaces. the in silico computation showed chemosorption, so tight sorption mechanism onto the clay surfaces. The cell adipocytes showed the PFAS adipogenesis toxicity was through metabolic and inflammatory pathways, and the clays was able to reduce the PFAS exposure as well as the cytotoxicity. The two living organisms showed the safety and detoxification of including clays in their culture media. And the rodent model showed that the clays in the oral gavage, they could reduce PFAS concentrations and the toxicities in the liver, serum, milk, and the reproductive systems in the adult, as well as moderately reduced developmental toxicity in the offspring. So collectively, the studies suggest that the clays have a notable potential for binding and detoxifying PFAS, suggesting the consumption of edible nutrient-amended clays could significantly decrease human exposure to PFAS toxicity from contaminated water and food. And lastly, I want to just show one of the ongoing studies in the lab, which is we want to include this edible clays in baby infant food and milk formula to reduce infant exposure to PFAS, because early infant exposure to PFAS is linked to increased risks of childhood obesity, later in their childhood. So, and you remember, we do see PFAS transfer in the breast milk and transfer to the offspring in the rodent study, and in the cells we show PFAS-induced lipid accumulation in the fat cells. So we first did a pilot study with collaboration with Dr. Johnson at Texas A&M University, where we added clays and carbons to cow milk. And we see that both carbons and clays could reduce PFAS concentrations in the milk. But carbon, they significantly reduced the protein levels in the cow milk, but the clays didn't show any significant reduction of the nutrients that we detected in the milk. And because of all of those preliminary results, we are now testing whether we could put clays in the baby food, in the milk formula, to reduce the PFAS concentrations and not interfering with any nutrients, and also validate their detoxification using these different models. So with that, I hope I conveyed my excitement about developing edible clays as a primary dietary barrier to block the uptake of PFAS and mitigate its downstream toxicity. And I would like to thank my collaborators, my students, as well as the funding support. And thank you all for your attention. I'm very happy to take any questions.
Thank you very much, Dr. Wang. Very interesting. And there are a number of questions. Let's start with some sort of basic things. How do you control which materials are absorbed by the clay or other sorbents so that they remove bad things and not good things?
Right. So
that's a general question.
Yeah, that's a good question. So there are the binding mechanism is quite a complex field, but there are a lot of parameters that we could control on the clays, such as their hydrophobicity on the surface, so we can target them toward more hydrophobic or hydrophilic compounds, but also we could adjust their pore size to only trap the right size of the chemicals. And there are also many other binding mechanisms. That's why we're having people in silico computation to delineate their binding mechanisms to better maximize the specificity of binding. But mostly for our part, one is the hydrophobicity, the pore size, and also we look at what amendments itself could have a higher affinity with this chemical. So we add those chemicals with higher affinity onto the clay. And then we have the in silico people maximize the, help us delineate the mechanism to maximize the specificity.
Is it possible to engineer the clay so that it would absorb other chemicals that we don't want in the body beyond PFAS, or is this approach really focused somehow on PFAS?
Yeah, that's a good question. So when we were doing the Superfund, we were actually trying to develop broad-acting clays to bind the kitchen sink. So because we want to remediate the environment, the kitchen sink. So yeah, at that point, we're trying to be as broad as possible. So we had a difference. For example, we created clays that are treated by the acid that simulate carbon structure, so they can bind almost a lot of things. But that's for the environmental remediation. But now that I move now target more on the human safety, really want to try to make sure that we only bind the bad side, the bad chemicals not interfere with nutrients. That's why I really try to increase the binding specificity. And that's why we have about 30 different kinds of clays. Some of them are targeting to be just very porous, like a sponge for environmental remediation. Some of them really target PFAS for specific binding mechanisms.
Thank you. It looked from the data that you presented that the clays in some cases seem to reduce the PFAS to a very, very low level, almost negligible, and in other cases reduced it substantially, but not all the way to zero. Can you comment on whether you see these clays reducing such chemicals to a level that avoids their harmful effects? Is that the appropriate target?
Yes. So yeah, they reduce PFAS to different extents because we use different matrix. If we're using the cell model, then we use DMEM, the cultural media for cells. And if we're using bioassays, then we're using their cultural media. If we're using the in vitro adsorption exosome, then we're using the simulated gut, the gastric or intestinal fluid as the matrix. So those different solvents and the different models, they will show different variations in binding to different extents. So the goal is to reduce to negligible. That will be the ultimate goal, to reduce as much as possible.
The work that you described, for the most part, was done in non-human models. What is the what do we know about the use of these clays in human populations?
Yeah, so for human populations, that is correct. For human populations, we have only tested the parent unamended montamoronite clays. So we did 6 human clinical trials in Texas and Africa using the parent montamoronite clay, and we show it's safe for human consumption. We didn't see any nutrient interference, and it was good for aflatoxin, not PFAS. Yeah, so for this newly developed clays, we're not at the human stage yet. You see me using different levels, hierarchies of, from cell to rodent, but yeah, not at the human stage yet for the newly developed sorbents. But I would love to. That's the ultimate goal, definitely.
Right, that's where you're headed. Would the FDA have to get involved in terms of approving the clays, or particularly approving the clays if you're adding things like carnitine or choline? Would the FDA have to get involved in approving those products for human consumption?
Yes, definitely. So FDA see the parent mammonic clay, and also most of the amendments that I added to the clay, they are all from the GRAS list, the generally recognized as safe list from the FDA. But it is definitely regulated. Right now, clays, they are approved to be included in animal feed as an anti-caking agent. That's why actually the parent clay, they're included in about 20% of the animal feed in Europe. But for humans, FDA has now approved it to be included in the human food. But you could buy Mediclay, which is the on Amazon, and we work with company who license with us for the clay technology, and they are the one, doing the work with FDA for all of that. And we just, work in the lab, make sure the science is solid. But yeah, it's approved for animals, but not human yet.
Yeah, and that means that some of the questions that are coming in from our audience probably can't be answered. So for example, one of our audience members is asking, what's the impact of clay ingestion in adults, children, babies on mobility in the GI tract? And what about adults taking GLP medications that slow down motility? But I'll bet those studies have not been done.
Yeah, for the parent clay, we have done some of those. So we know that a human eating parent clay, if you eat it more than 10, if you add parent clay at levels higher than 10%, then that could cause constipation. That's the biggest side effect that we see in human studies. But other than that, we don't see any side effect from the human studies fed with clays. Yeah, other than constipation, we didn't see anything else. We, when we did a human study, we had CDC and USAID on site, and they classified the clays as minimal effect in humans based on the results. And yeah, that's why, this clay we think is promising. So we keep doing it and now targeting PFAS.
If these clays were to be administered in human populations, how and where would it be delivered? Would it be in a pill form, in a beverage of some sort, as a food additive, as we do with vitamins and other things? What are your thoughts about how this might be applied eventually?
Yeah, that's a good question. So when we did the human clinical trials, we use the different delivery format. We have done pills, capsules. We have even included clays in cooking condiments, so we could cook with it. We have also used sachets like tea bags to be included in flavored water, and we have also just directly mixed it with the porridge that people drink in Africa. So the efficacy remained the same regardless of the delivery formats. So we think, all of those format you said, they could all work. And right now, all the studies I have, they are for short term. That's why I'm really focusing on the susceptible windows of exposure, pregnancy, lactation, developmental of newborns, to make sure that at least those most susceptible windows are my target for now.
Towards the end of your presentation, you said you were starting to look at using these clays in humans, and particularly in pregnant women and nursing women and young infants. That seems to be a particularly high risk population for initial human work. Can you comment on that?
Yeah, so I'm not directly recruiting pregnant women or nursing women or infant directly in my study. So that will still be a in vitro and in vivo study first. So what I mean is that I'm using the still using the matrix. For example, I'm using infant food, I'm using infant formula, I'm using breast milk. So those are my samples for studies. I'm not directly recruiting people yet. So
Okay.
yeah.
Okay. All right. Thank you. That wasn't entirely clear. Does the moving in a different direction, does the body store PFAS like the body stores other toxic materials like lead, for example? Does the body store PFAS and if so, where?
Yeah, yeah, it does. The body does accumulate PFAS, especially the long chain, to different extent. I believe the long chain, like PFOS, could the half-life, I think it's years in humans. And where do they stay? I think mostly in the liver. I've also heard saw papers showing PFAS in the bone marrow. Um, so I would think mostly in the liver. And also pancreas, I think I saw that somewhere too.
Now, will the and you may have done this in animal studies, not humans yet, of course, but will the clay remove the stored PFAS? Are you looking at whether it really is reducing PFAS throughout the body, or are you focused mostly on levels in serum or in fat tissue?
Yeah, so I think that's a good question. So the primary design of using the clay is using it primarily as a prevention. So that's why I included PFAS in the gavage that is contaminated with PFAS. So it's like co-exposure, binding in the GI tract. And the beauty of using clay is that our body do not take clay. So the clay is not gonna get to the blood, to the organ. So the clays, they're gonna bind PFAS in the gut, and then they're gonna be directly excreted through fecal elimination. So that's why the major binding sites is the gut for prevention. But because PFAS goes through this intrahepatic recirculation, after PFAS exposure, they could return to the gut. And because the gut is our binding site, then so if they return to the gut, then that leaves window for the clay to be effective as a post-exposure treatment. But if they are, stay in the serum, stay in the liver, then the clay won't get to there because we want it to be, directly get it out.
Right. That makes sense. That makes sense. Let's see. One of the questions that we often have with new treatments is whether subgroups in the population might have an adverse reaction where most of the population doesn't. And I recognize that you're not there yet in terms of human populations, but is there any indication from the animal work or other work that there might be subgroups that might have an adverse reaction, or is it generally just been positive across the board?
Yeah, that's a question I thought of myself also. But yeah, that's why when we did the human clinical trials, actually one of the 6 human clinical trials we did was with children from age 3 to 9. And again, we didn't see any side effect in the clay administered groups. And we are actually targeting the susceptible groups like the children, the pregnancy. And based on my animal or the previous human studies, we didn't see any subgroup that are adverse effect. And actually, pica, that's human eating disorder. And clay is a very common form for pica. And actually, pica happens most commonly in children and pregnant women. And in Africa, still these days, a lot of pregnant women in Africa, they intentionally seek to eat clays because that's part of their culture, their culinary habit to eat clays as a toxin binder to supplement nutrients. So actually we see those subpopulation most vulnerable, but they tend to, the clays actually protect them more, or at least that's our target, to protect them. So we don't see any subgroups that are just absolutely not suitable for the clay.
Right. It's interesting that you have these populations that naturally ingest some of these clays so that you can look at those kinds of things even without having to provide the material to people. We are out of time, unfortunately, even though there are more questions. So let me wrap things up for today. I want to thank Dr. Wang for a wonderful presentation, and I want to thank our audience for very good questions. As I mentioned earlier, we will be posting a recording of today's webinar on our website in the next few weeks. I want to thank everyone for joining today's lecture.